Welcome to this explainer. Let's frame the human body for what it actually is, a highly contested microscopic ecosystem. Literally right now, your immune system is constantly evaluating self versus non-self, making split-second decisions that keep you alive. Today we're going to break down exactly how that works. But more importantly, we'll look at what happens when the delicate balance of our microscopic allies gets shattered. We are bridging the gap between basic immunology and clinical pathology to get you ready for the wards and, of course, the boards.
Here is our roadmap for today's clinical lecture. We're moving fast through antigen foundations, T-cell dynamics, flora and symbiosis, normal flora shifts, pathogen immune suppression, and wrapping it all up with a quick exam revision summary.
All right, Section 1, Antigen Foundations. Let's set the rules of engagement.
Let's clear up a classic board trip-up right out of the gate. An antigen is just any substance that elicits an immune response and reacts with the products of that response, like antibodies. But there is a super crucial distinction between antigenicity and immunogenicity. Antigenicity just means a substance can physically lock onto an antibody or a T-cell receptor. Immunogenicity, though, that's the actual power to kick off the whole immune response in the first place. So remember this absolute rule. While all immunogens are antigens, not all antigens are immunogens. Some things can bind to immune products without actually having the juice to trigger their creation from scratch.
Take a look at the concept of valency here. See that multivalent antigen? It is basically bristling with multiple antigenic determinant sites, or epitopes, poking right out of its surface. Because it has all these multiple binding sites, a single foreign molecule can interact with a whole bunch of antibodies all at once. This physically generates a massively stronger immune reaction compared to a monovalent antigen that only has one single epitope to offer. Basically, size and structural complexity matter. The larger and more complex a molecule is, the more potent it is as an immunogen.
Now, let's tie this basic science directly to the warts. Ever heard of haptens? These are tiny, low molecular weight substances that are perfectly antigenic, meaning they can react with antibodies. But they are definitely not immunogenic on their own. They're just too small to trigger the alarm. To provoke a real immune response, a hapten literally has to hitch a ride by chemically coupling to a larger carrier molecule, like a protein in your own body. The classic clinical example of a hapten is penicillin. By itself, it usually flies right under the radar. But if it binds to host proteins, boom, the immune system suddenly recognizes this new complex as completely foreign, and that triggers the allergic reactions we see so often in patients.
Moving on to Section 2, T-cell dynamics. How do we actually mount an attack?
You've got to draw a hard line between these two mechanisms for the boards. T-dependent antigens, which are mostly proteins, absolutely require the assistance of T helper cells to stimulate B cells. And this teamwork is highly effective because it leads to high-affinity antibodies and critically generates memory cells for long-term immunity. On the flip side, T-independent antigens, like the polysaccharides you find in bacterial capsules, can stimulate B cells directly without any T-cell help at all. But there is a massive catch here. This independent route usually results in a much weaker, mostly IgM-based response, and, worst of all, it completely fails to produce those crucial memory cells.
Now, superantigens are a whole different beast. Normally, an antigen gets carefully chopped up and presented inside the little groove of an MHC molecule. But superantigens, they completely bypass that normal intracellular processing. They are bacterial proteins that just latch directly onto the outside of both the MHC Class II molecule on the antigen-presenting cell and the T-cell receptor. This causes a massive, totally nonspecific activation of T-cells. We're talking a dangerous, systemic release of cytokines. It's basically an immunological short circuit, and it can throw a patient right into severe shock.
Okay, Section 3, Flora and Symbiosis. Let's talk about our microscopic allies.
Think about the sheer density and anatomical distribution of your normal flora. While your internal organs, like the heart and lungs, are strictly sterile, the parts exposed to the outside world are literally teeming with life. And different regions support entirely different microbial communities, based on stuff like moisture, pH, and oxygen. The skin is dominated by bugs like Staphylococcus epidermidis, but as you move down into the gastrointestinal tract, the numbers just explode. Down in the large bowel, bacterial density reaches a staggering 10 to the 11th power per gram of tissue. The vast majority of those, anaerobes like bacteroids.
We didn't just passively tolerate these bugs over millions of years, we evolved with them. This host microbe evolution has created strict mutual metabolic dependencies. Your commensal microbes are actively protecting you right now.
In the reproductive tract, vaginal lactobacilli ferment glycogen to keep the pH nice and acidic, which actively prevents yeast and other pathogens from overgrowing. In the gut, your normal flora pumps out metabolic waste products and specific antibacterial factors called bacteriocins that suppress invaders. Plus, just by existing in such crazy numbers, they occupy almost all the available ecological niches. There is literally no room at the end for potential pathogens to set up shop.
Which brings us to Section 4, Normal Flora Shifts, the clinical betrayal.
Here's a pivotal clinical question for you. We spend so much time fighting pathogens, but what happens when our own medical interventions inadvertently wipe out our first line of microbial defense?
Well, the reality is that broad-spectrum antibiotics, while totally life-saving, can be ecologically devastating to a patient's gut. Take a drug like clindamycin. It can drastically wipe out that protective flora. When those good bacteria die off, they leave behind empty ecological niches. You've just lost your colonization resistance. Suddenly, an opportunistic organism like Clostridium difficile, which naturally resists the treatment, wakes up and finds itself with unlimited resources and prime real estate. It proliferates like crazy, releasing toxins that cause severe antibiotic-associated diarrhea or even life-threatening pseudomembranous colitis. Our intervention literally paved the way for the infection.
Section 5. Pathogen immune suppression. Active sabotage.
This dichotomy is absolutely fascinating. Your commensal bacteria guard the ecosystem passively. They occupy niches, produce fatty acids on the skin, maintain that acid pH. But true pathogens? They actively dismantle your defenses. Look at certain viruses. Measles actively infects monocytes and downregulates the synthesis of interleukin-12, which severely depresses cell-mediated immunity and leaves the patient wide open to secondary bacterial pneumonias. HIV goes straight for the generals of the immune system, depleting CD4-positive T cells through rapid viral replication and by inducing cellular apoptosis. One protects the house, and the other actively burns it down from the inside.
Finally, Section 6, Exam Revision Summary. Let's synthesize this high-yield info. All right, take a mental snapshot of this master review table. You need to keep the strict immunological definitions on the left totally separate from the broad ecological relationships on the right. On the antigen side, exogenous antigens come from outside, endogenous are generated within, haptens require a carrier protein, and superantigens bypass normal MHC processing to trigger dangerous cytokine storms. On the symbiosis side, commensalism is where the microbe benefits harmlessly, mutualism is where both rely on each other, parasitism is where the host actively gets hurt, and dangerous flora shifts are what open the door to those opportunistic infections. Keep these categories locked down for the exam.
I want to leave you with a really provocative thought on clinical stewardship. As we increasingly rely on routine, broad-spectrum antibiotics and advanced immunosuppressive therapies, we are fundamentally altering the microscopic environments within our patients. Are we permanently altering human evolution by changing these ancient host-parasite relationships? By saving lives today, are we inadvertently selecting for much more resilient, opportunistic pathogens tomorrow? It's a huge question and something every single future clinician has to ponder as they write their next prescription. Thanks for joining me on this explainer, and keep questioning the microworld around you.